Hello, my name is Ali Askari, and I am an undergraduate researcher at the University of Texas at Austin. In today's presentation, I will talk about Aptamer and Aptamer database. First, let's start with what is an aptamer. Aptamers are oligonucleotides that binds to a target with a high specificity and affinity. This target could be proteins, viruses, bacteria, cells, tissue, nucleotides, almost any small molecule. This characteristic provide aptamers the wide range of practical applications. Some examples of these practical application areas are diagnostic kits, medication, and industrial use. With a staggering increase in the rate of aptamer discoveries and research, there is a high need for creating a comprehensive and sustainable aptamer database. I am presenting to you the long-term preservation and collection of aptamer sequences in a dataset available via an online database. Long-term preservation of aptamer sequence refer to a publicly available dataset so everyone may access this data and build upon it. And a long-term collection refers to the implementation of this project in the Aptamer research course. So new undergraduate researchers are constantly being trained to update the dataset. Let's briefly talk about how we collect aptamer sequences. We utilize PubMed to gather publications that found aptamer sequences. After collecting the necessary information in our dataset, a reviewer would go over an aptamer sequence entry to confirm its accuracy. Then the dataset would be transferred to our database platform, which would be accessible via our website. In Figure 1, you see an aptamer entry would look like in our dataset. Figure 2 is our search engine, and Figure 3 is our result page. We would hope this database help researchers answer some questions such as what type of nucleotides are found in most aptamers? Are there similarities between aptamer sequences that was selected against related proteins? And many more questions. In the future, the database research project will continually work toward collecting and peer reviewing information to encompass all aptamer sequences, as well as performing data analysis and interpretation of our data. As of today, this database has 1,417 aptamer sequences completed, which means they have been peer-reviewed at least once. Upon publication and release of this database to the public, this would be the largest aptamer database in the world. Now, let's briefly go to our website. Here, the search engine includes the year of the paper that was published. The aptamer composition can be searched by target, aptamer sequence, possible application, pool, serial number, and keyword, any keyword that you have in mind. Here, I'm going to go for a target HIV. The result page is organized for a quick glance to our data. This quick glance includes the year, the target of the aptamer, and the aptamer composition, and the sequence, affinity, possible application, citation, and corresponding author. We can expand the sequence to see the full sequence of the aptamer in here, or compress it. For more detail, we can click on View More Detail and see more information about each aptamer. At last, we have been given a comment section to submit any comments or questions or any concern regarding each aptamer sequence entry. At last, I want to thank you, all of you, for listening to me. Please don't hesitate to reach out to us. Thank you.